B cells contribute to adaptive immunity by secreting antibodies, and the response of B cells to an injected immunogen is usually measured by analyzing the specific antibody produced in a humoral immune response. This is most conveniently achieved by assaying the antibody that accumulates in the fluid phase of the blood or plasma. Such antibodies are known as circulating antibodies. Circulating antibody is usually measured by collecting blood, allowing it to clot, and then isolating the serum from the clotted blood. The most important characteristics of an antibody response are the specificity, amount, isotype or class, and affinity of the antibodies produced. The specificity determines the ability of the antibody to distinguish the immunogen from other antigens. The amount of antibody can be determined in many different ways and is a function of the number of responding B cells, the rate of antibody synthesis, and the persistence of the antibody after production. The persistence of an antibody in the plasma and extracellular fluid bathing the tissues is determined mainly by its isotype. Each isotype has a different half-life in vivo. The isotypic composition of an antibody response also determines the biological functions these antibodies can perform and the sites in which the antibody will be found. Finally, the strength of binding of the antibody to its antigen in terms of a single antigen binding site binding to a monovalent antigen is termed its affinity. The total binding strength of a molecule with more than one binding site is called its avidity. Binding strength is important, since the higher the affinity of the antibody for its antigen, the less antibody is required to eliminate the antigen, as antibodies with higher affinity will bind at lower antigen concentrations. All these parameters of the humoral immune response help to determine the capacity of that response to protect the host from infection.